Good morning, viewers, and welcome to today's session, which I want to try to explain to you some important issues here. Like we are talking about membrane, proteins, DNA, and gene expression. This is the chapter that we've been going with. It is 2B. We have looked at 2A. So today we want to talk about specifically proteins and the DNA. And an example of the protein that we want to look at is called the enzyme. Now I want you to remember that in the study of the biochemistry, it's important that we realize that there are reactions that take place inside the cell. Now these reactions normally make cells get important things like the energy that they need and formations of new materials. Now these reactions, in most cases, they are very slow. Why are they slow? Because the normal body temperature of a human being, for example, is too low, is 37. So at that particular point, the reactions might be too slow, such that they might not meet the need of the cell. So it is important that those particular reactions in the cells are made quicker. So what makes them quick is what we are calling the enzymes themselves. So enzyme is a protein. And we know that proteins are made up of simple molecules like amino acids, which are joined with a peptide bond so that they can form a very long chain. So enzyme is also in that particular manner. So this particular chain of the proteins, they can be joined so that they have got long chains that forms structures. So the structures can be a secondary structure, it can be tertiary structure, or the quaternary structure. Those are some of the structures that we have seen. So that is what normally that is what normally happens. So those structures are formed due to formation of the bonds like the hydrogen bond, disulfide bonds, and the ionic bond. So that is what we have seen sometimes back. So today I just want us to understand what happens, what is an enzyme. What is an enzyme? We want to really try to understand what enzyme is and how does it help us in daily activities. Now enzyme, we can refer to it as enzyme is a biocatalyst which speed up a reaction or a chemical reaction in the body. Chemical reaction in the body. So it is a biocatalyst that is speeding up a chemical reaction that is happening in the body or in the cells of a living organism. So enzymes are based on the cells of the living organism. So we'll be seeing how this particular catalyst reacts. Now I want you to understand that a catalyst, for example, a catalyst, we should understand what a catalyst is. So a catalyst is a substance that changes the rate of reaction without affecting the product, and in the process itself is not changed during the reaction. That is what a catalyst is, a substance that speeds up a rate of reaction, and itself it is not changed at the end of the reaction, and then it does not interfere or it does not change the product that is formed there. That is what a catalyst is. So the enzyme is an example of a catalyst, only that this one is a biological catalyst, a living catalyst. So that is what normally happens in it. Now, like I've said, that in most of the reactions it might be very slow for the reaction to take place. So this one here will make the reactions to be a little bit quicker in the cell, so that the cells can be meeting the need. The requirements that it needs, it can easily meet them in time. So we will be seeing this one in either the cells of we have those particular enzymes that can be working inside the body, or they can be working outside the body. So how do the reactions normally occur? Now I want us to understand that we have got two types of reaction. There are types of reactions in the cell. Type of reactions. So we have got the reaction that help us in building of new materials, like it is forming materials. So we can talk about reaction one, that type of reaction that help in the building up material, we call it anabolic reaction. So what is anabolic reaction? This is a type of reaction that help in the building up. So we can sometimes call it build up process, building up material. So this is one reaction. It will need enzymes to form new materials. Then we also have got a reaction whereby we break large molecules into simple molecules. So that one is called catabolic. This one is called catabolic reaction. So these are the reactions that will involve the use of enzyme. This one we say is a breakdown. This is breakdown of materials. So you see there's two things here. We can need to break down materials, especially for example during the digestion. We can break them down so that they can become small soluble materials that the body can transport. So when they are being transported to the sites where they are needed, that process should be quick enough so that they can give us whatever the body needs. Now when we are forming materials, we also need to form materials quick enough so that they can help us in perfecting the functions of the body. So these are the two types of reactions that are there, and these reactions can only be made perfect when we are facilitating them using a catalyst that is called the enzyme. So all these reactions are the metabolic reactions. These two reactions make up what we are calling metabolic reactions, which will be leading to the body metabolism. So the path that is being followed to achieve this is called metabolic path. That is what we can refer to them. So this is what enzyme is trying to, this is what is happening within the enzyme, the function and the meaning of the enzymes and the work that it should be doing for us. So that is one thing that I really want us to understand. Now I have really mentioned that these enzymes, they can be working inside the body. So we can be, I want us to look at the naming of the enzyme, naming of enzyme. And in this, I want you to understand that we have got enzymes that are working inside the cells where they are produced. So we can call that intracellular enzyme. This is an enzyme that is produced within a cell and then it works there. It is really working in that particular cell. So that is intracellular enzyme. An example is what is called the DNA polymerase. This is an enzyme that is produced within a cell and it is working within that particular area. So that is an example. Then we also have got the extracellular enzyme. Extracellular enzyme. Now, there are those particular organisms that their digestion is outside their body, actually outside the body. A good example is like the flies. We know that the flies normally will release enzyme on the substrate they want to digest outside their body. They break it down and then they siphon it in so that they take it in already simple form. Mushroom, like the fungi, is also one example. And even the digestion in the human being, it is done outside the cell. So it is an extracellular, the human being can also produce extracellular enzyme that is produced in the glands and then they are released in the digestive system to break down this particular food. So that is an example of the enzymes that we have. So how do we give this particular enzymes name? How do we name the enzymes? So this is how the naming of the enzyme is done. So we are going to follow some three basic rules in naming of this particular enzyme so that we see the names that we can give to them and according to what particular principles that we name. So we can be giving the enzymes a relatively short recommended name. So we can give them a relatively short recommended name. Recommended names. Like in this case, these names will be based on the substrate. So we are naming them based on the substrate. So naming is done based on substrate they work on. Substrate they work on. So that one means that the enzyme is going to derive its name from the substrate that they are digesting. I can give a simple name that you have already known. So assuming that we have got a substrate like a starch, an example of a starch like amylose. amylose. This is a substrate. So we can get the name of enzyme from here by substituting that with this suffix here, so that the enzyme become amylase. So you see, this is a very short name, and it is based on the substrate that the enzyme is working on. So that is one way by which we can be giving the enzymes the name. Then we can also give them long systematic name based on the process that they are working on. So you can have a long systematic name based on the process. Based on the process they are working on. So that is another way by which we can give the enzyme name. Like, for example, there is this process of producing energy, and when energy is being produced by respiration, it's given in form of a molecule called ATP. So we can talk about ATP, adenosine triphosphate. So this is the molecules that give us energy. So the enzyme that will be working on this will be called adenosine. We'll be calling it adenosine phosphotransferase. Adenosine phosphotransferase. So you see, this is an enzyme that is based on a process of energy production. So we can be giving the enzyme's name based on the processes that they are working on, or the substrate that they are working on. So those are some two clear ways by which we can be giving the enzyme name. Or we can be classify them in terms of numbers. So we can talk about classification by numbers, so that we just number them up. Like we can be talking about, example is EC 2.7 or 3.2. So these are examples of ways by which we can be giving the enzymes their name that we'll be talking about, and we'll be referring to them in that particular manner. So this is what I really want us to understand at this particular point, and I want you to really realize that when we are referring to enzyme, it is a protein substance, protein in nature. So anything that is going to affect the proteinous substances, or they are going to possess the protein properties, that one means that whatever is going to affect the protein will be affecting the enzyme, because they are having that particular protein in nature. So that is one thing that I really want us to understand in the introduction part of the enzyme. So thank you very much. I want us to continue so that we may see, after looking at the enzymes, the introduction of the enzyme, I want us to see how the enzyme do work. That is what I want us to see. And I want you to realize that enzymes will only work when they are reacting with the substrate. And normally for the substrate to react, the substrate needs to accumulate energy so that it can break the bond of the molecules that are holding them. Now for that one to happen, there must be an energy that's accumulated to help us to break this solar bond so that a reaction can start. That particular energy is called an activation energy. Now it can be very, very enormous, can be very, very big when it is not facilitated. So the enzyme as a catalyst is really helping us to lower that activation energy, so that the molecule that is reacting does not need to accumulate much of energy that will be helping it to break its own bond so that a reaction can start. So like I can show it to you here using the energy path reaction here, so that we can look at the initial reaction. We have got activation energy A. This one is happening whenever it is not catalyzed. So you can see how we have got an uphill here of an energy being accumulated to break the bond before the reaction starts. So we have to break the bond of the chemical so that the reaction can start. So that is the activation energy. So you can see how large it is, what amount of energy is needed if any case the reaction is uncatalyzed. But when we use enzyme is trying to lower the energy required for the molecules to break down the bonds of the molecules to be broken down so that a reaction can proceed. So you can see how the energy is low. So at the activation, the reaction will be very quick because we have got a low energy that is required because the process is being catalyzed. So that one will be giving us the final product there. So that is one thing that we should really understand, the benefits of having a catalyst, which in this case is an enzyme. So it is going to lower the activation energy that is required. So it makes the reaction to become very, very quick. So that is one bit that I want us to see. But into details, I want us to see how do this particular one work. How is the enzyme going to take part in the reaction? So that is the question that we may ask ourselves. How do enzyme work? How do enzyme work? So we can ask ourselves this. So number one thing that I want us to note is that as much as the enzyme is a molecule, a protein molecule that helps us in the speeding up the rate of reaction, this molecule is actually having a site. So maybe I can indicate it here like this. This is the enzyme. There is a site for it here. So I can refer to this as the enzyme in this case. I can demonstrate that. And then I can have the molecule that it's working upon, which in this case will be this one here. Let me call this the substrate. The substrate. So you see this particular thing, how they look like. Enzyme have got one side that is having a shape that can make the substrate to fit in. So this is the site here. So we can call this active site. Actually, this is where the reaction will be taking place. So this particular substrate will be fitting in here. So it's more of a, the enzyme is going to lock itself with the substrate. So that particular theory we can be referring to it as lock and key hypothesis, whereby the enzyme is like our lock, the substrate is our key, which is fixing itself onto the active site, and they combine together. So we can see that this process will be giving us a picture that looks like this. At the end of the day, we'll be having, this is now the enzyme again, and then the substrate now comes in and it fits itself here. This is now my substrate. You can see how it is fitting there. This is the substrate that I had. So these two here now will be forming the enzyme substrate complex. So now we have got the enzyme substrate complex. So the two combine to give us a complex molecule like this. So at this point here, the reaction is taking place at that particular active site. And I want you to remember that not every substrate will be fitting on the enzyme. So enzymes are very specific. So they have got a definite shape that only a specific substrate can fit in. Therefore, we can say that enzymes do select. They are selective. So they are very specific to which substrate they are working on. Like for example, if my substrate was like this, if in any case this was my substrate, you can see there is no shape here that can fit on this. So if these are two substrates here, you will be seeing that this enzyme here will only be selecting this to work on, and this one will remain uncatalyzed and reacted upon. Why? Because the shape that the active site that is having here is not specific to the shape of the substrate that we are seeing at that other point. So this one will not be broken down, and this one will be broken down. So at the end of the day, we'll be seeing that when this thing has been simplified, has been broken down, we'll be having the enzyme now releasing the substrate here, the product at this point. So we'll be having our enzyme with its shape remaining there. It's unlocked, so that it has got the shape, the initial shape that it was having. It unlocked itself, but it is giving us a simplified product. Like this is now how the substrate is now. They have been simplified. So this is the product. So you can see we are breaking it into two simpler molecules. So if this was food, at this point you can see that it is simple and the absorption can take place. If this was digestion that was taking place, so you can see this is what happens. So at this point we can say that enzyme work by locking itself with substrate. they lock themselves with the substrate on a side that is called active site. So that is very important for us to note that in the action of the enzyme, we are going to be referring to the active site as the point where the reaction is going to take place. Every enzyme have got an active site with various shapes just to fit on the specific substrate that they are going to work on. That gives us a property of an enzyme, that they are substrate specific. So they are going to digest a very, very specific substrate. And then the moment the reaction is completed, the active site is set free so that it can continue. So this reaction, the action of enzyme, is actually based on the active sites. And in any time we are explaining this in the exam, this one will be our clear reference point. So that is the thing. Now I want us to continue and understand another idea here, that however much the substrate is specific, like however much the active sites can be specific, it is also worth noting that these particular active sites, they are not fixed. What do we mean? Now this is what we mean. This particular shape here, they are flexible, meaning that they can adjust to accommodate the substrate, but their adjustment is limited based on that specificity of their site. Like for example, if I have got an enzyme that have got this type of active site, like I have this is my new enzyme that I'm referring to. So this is my enzyme. This is now the active site. You can see the shape that it is having here. If I have got this substrate here, what will happen? If this is my substrate. Now, if you look at my substrate, assuming that this is the substrate that I have. Now, if you are keen enough, you will notice that that substrate still can get into that particular active site. It can get in. But when it gets in there, it will force this enzyme here to adjust. It is flexible. So it can adjust to accommodate that, because that thing can fit. So if it can get in, it is the adjustment that of the enzyme that will be helping it to make sure that it works on this. So suppose they now form an enzyme substrate complex. What will I have? Excuse me. This is what we'll be having. You will see that, you notice that the enzyme now, that appearance is still there, and then there is that adjustment so that those sharpness that we are seeing, they are not there, so that the enzyme, the substrate here, you can see the substrate is being accommodated in that manner. So this is now the enzyme. We are getting the enzyme substrate complex. Remember, these are protein. Enzymes are protein nature, so it can adjust so that it fits itself in that particular shape that we are having. So this one will be able to work on this particular enzyme. Then we can have, at the end of the day, the substrate being released. The product now still will come in, but the shape after the releasing, this is our shape that will keep on remaining with us as we had it there initially. So you can see when it was here, this is what we have. It adjusted to fit on the substrate. This is the substrate. So the two here forming the complex, and then it's going to release for us the product of the substrate. These are the product, and this is the enzyme. So this one here is enzyme that is fitting itself with the substrate. So this is called induced fit hypothesis. So this is what we are calling induced fit hypothesis. The other one was simple lock and key hypothesis, but this one now we are inducing it. We are making it to happen. We are forming it by ourselves. So the enzyme is adjusting. So that is one thing that I really want us to really note, that the enzymes are there, as much as they have got the active site that are very specific. It is not rigid, such that the substrate that is of its nature cannot fit, because these particular shapes that we are having here, they can adjust, the enzymes can adjust so that it can accommodate it. So this is how the enzyme is working for us. So that is the thing that we should really note here, so that we can see how the enzyme works. So that is the working of the enzyme. So we have seen how the enzyme work, whether it is induced or the lock and key hypothesis. You have seen how both cases can be helping to facilitate the rate of reaction. Now I want us to continue and confirm this, that is it true? We want to see how the evidence for the relationship between the structure and the function of enzyme. So we want to see how the structure of the enzyme affect its function, and what are some of the factors that can affect the structure, and how will that one be affecting the functioning of the enzyme. Now before we go into that, let us remind ourselves of some of the facts that we have known about enzyme. Number one thing that we have known about enzyme, that the enzyme has got an active site. That is the first thing, which is a small depression on one side of the enzyme. That particular depression has got a specific shape which the substrate will be fitting into. So that is one key thing that we should be knowing. So not every substrate will be fitting into that side, but specified one will be fitting in there. Number two, we have really known that enzyme is protein in nature. Therefore, anything that is going to interfere or affect the proteinous substances will be affecting the functioning of the enzyme. So if any case we can interfere with that particular active site, we will be interfering with the reaction. So the reaction can either stop, or it cannot even proceed at all. So those are some of the facts that we have been looking at about the enzyme. Now we have also known that the enzyme, they do not change, or they do not contribute to the change of the product. So in other words, enzymes, they only facilitate the reaction, but they don't take part in the reaction. They remain unchanged. So that product that is coming out is just that product, and the enzyme is not going to affect that particular product in terms of how it is, but it is their numbers that is going to help us. The number of the product that's going to come out, that's what it can facilitate, the speed, but not what comes out of the reaction. So that is one thing that we should also remember here. So there is no effect on the modification of the equilibrium and the product that is coming out from the enzyme. Those are some of the things that we should actually remind ourselves about. So let us look at some of the things that the evidence for the relationship between the structure and the functions of the enzyme. Now we have seen that when the enzymes react with when we have got enzyme reacting with substrate, what normally comes out is enzyme substrate complex. That is what we saw. This is one thing that we saw. And then finally, we will be giving this one here, will be giving us enzyme and the product. So if we take this one out, if we take our enzyme out again from this enzyme substrate complex minus the enzyme, you'll be getting enzyme plus the product. Those are the things that we are seeing here. So in any reaction, suppose I have got the enzymes here. Let us look at this little bit. If I've got enzymes, I can be having substrate. So if these are my enzymes, assuming that I've got enzyme here, these are my enzymes, these are my enzyme. So I can be having substrate here. So what do I really want? I want to break down this particular substrate. You can have a look at how substrate molecules are here. So what will happen? This substrate here will be combining with the enzyme, the substrates, and then at the end of the day I'll be getting product. That is what I am having. So in my reaction, if any case this is how my reaction will be happening, I will be having the maximum reaction. If I have to illustrate this on a graph, so I can be having V maximum reaction. So this one here can be substrate, and then I can be having rate of reaction. This is what I'll be having. So if I was to illustrate this in a graph form, I'll be seeing that my graph will be looking like this. So the reactions here, if the substrates are coming in here, the rate of reaction will be made quickly. So it is going to happen very, very quickly until it is approaching the maximum point here. So the shape will be like this. The maximum when it's approaching the maximum point. Why does it behave like this? Now this shows that at this particular point here, I have got free enzyme which will be fixing themselves with the substrate to give me the product. So again, I am referring to the active sites being involved in the reaction. Much of them are involved in reaction, and some of them are still free to continue with the reaction. So the reaction is becoming faster. When I reach at this point, why is it that I am not getting any more product coming in, the number increasing beyond this? It is clear that at this particular stage here, what has happened is all the enzymes that I'm having now, they are involved in the reaction. So there is no free active site that is bringing in a new product, a new number of products coming in. So at this point here, we are seeing how the active site is related to the number of the substrate. So we can use some of the factors to really prove that there is that particular effect on structure and the product that is going to come out, which is the function of the enzyme. So we can look at it in terms of the first one we can check on is temperature. Maybe we can use the temperature as one of the things to confirm this. So I can talk about the temperature here and see how this as a factor can have influence on the structures and then the functioning, the entire functioning of the enzyme. Now this is one thing that is very, very clear. If the temperature is really going to increase, suppose I have got my temperature being from, let's say, 10 degrees Celsius. The human body temperature is constantly at 37, for example, 37 degrees Celsius. So you will realize that we can have ranges of temperature, like let's say from 10, we can go to 20, we can go to 30, we can go to 40. These are ranges of temperature. Now, do we really think that the way the temperature is here, the reaction might be the same when it is at 20? The answer will not be like it is not going to be true that way. The reactions will vary because there is what the input that the temperature is going to have on both enzyme and substrate at various temperatures, so that the reactions, the speed of the reaction will change entirely. Like look at it in terms of this one, in terms of the diagram I have here. If this is the temperature, if this is the enzyme, this is the substrate, they should be randomly moving so that they meet one another. That is the first thing. There must be movement so that they have the collision for a reaction to occur. Because reaction will not take place if there is no meeting, if there is no collision. So what makes them to move? Is there some individual moving them? The answer is no. These things are moving on their own. They have got random motion. And that random motion is because of the energy that they store. So we can have that they have kinetic energy. That is both enzyme and the substrate. So the two of them have got energy that is going to make them to move and meet. Now this energy is influenced by the temperature that is available. So if we are increasing the temperature, it is proportional to the kinetic energy. The kinetic energy will increase. So we can say increase in temperature, increases kinetic energy, increases kinetic energy of molecule. So when the molecules are being increased, the energy in them is being increased, you can see that the rate at which they are going to collide is also going to be increased. And when they are increased, the speed of collision is also increased. So that one means that the rate of reaction will be improving with the increase in temperature. That is one thing that I want us to really understand, that the increase in temperature will be increasing the rate of reaction. But this one is always moving up to a limited point. It cannot just happen throughout. So when we exceed the temperature that is required, like for example the body temperature, this parallel reaction will stop, because increase in temperature that is beyond the required one, the optimum, will mean that the active sites, the shape will change, and that change in the shape will make the reactions to stop, so that most substrate will not be fitting in that parallel shape, as we have learned before. Therefore, that one means that the reaction will stop, and at that point we'll say that the enzyme is denatured. And in this particular denaturization, we are talking about the change in the shape. So that is one thing that is there with us. So that is how the temperature is actually affecting the rate of the reactions of the molecule. Now, with this particular increase in temperature, it can always be expressed as a temperature coefficient, this rate of reaction. And we can have an equation that is helping us to understand that. We can have a temperature coefficient of this being equivalent to rate of, we can talk about rate of reaction, which in this case we can have x plus a 10 degrees Celsius, meaning that in every increase of 10 degrees Celsius, the rate of reaction will be increasing by 2. So that is one thing that we should be understanding here. So this one will be over rate of reaction at temperature x degrees Celsius. So this is how we can express that increase in rate of reaction in relation to the increase in temperature. So we can be expressing it in terms of that particular equation. So that is how the temperature is really actually affecting that. So in the explanation again here, there are some two things that is very clear for us. That temperature is affecting the kinetic energy of molecules. That is one thing that is very clear. When the kinetic energy of molecule is affected, the speed at which they are moving and meeting one another, that is enzyme and the substrate, will be changed. So if it is increased, then the rate of movement will be quickly, and then that collision will be quick, and that one will mean that the reaction will be increased up to a limiting point whereby the active site is not changed. When it is changed, that is being denatured, it means that the reaction will finally stop. So that is how the temperature is affecting the rate of reaction. And this one can therefore be expressed in a graph, like for example, when we can talk about the human being temperature, which is known. We can be having enzyme like, suppose I express this in a graph. So I will really see that the reaction when the temperature is low, it's like that. But when the temperature is increasing, it goes up. When the temperature is increasing beyond, it stops. So this one means that up to this point, this is my optimum temperature. Optimum temperature, the best temperature. So the rate of collision between the substrate and the enzyme molecule are so quick. Therefore, the rate of reaction is very high. What happens at this other end? The enzyme, the active site of the enzyme is destroyed. It is denatured. So the enzyme can no longer meet with the substrate so that they act. So that is how we can really express this. And another thing that is worth noting is that not all temperature have the same optimum temperature. The optimum temperature will depend on the organism. Like the normal body temperature of a human being is 37. So most of the enzymes that work in the human cell, their optimum temperature is 37. We can have some organisms like the plants, the desert plants, the temperature that they can tolerate is a little bit higher. Therefore, it means that their enzymes will be having more optimum temperature, high optimum temperatures compared to these ones. In the industries, we can have the artificial enzymes that have got their temperature, optimum temperature being increased. So this as a factor will also depend on the type of the enzymes and the organism that we are referring to. So that is the thing. Factor number 2 that I want us to consider, I think this one can be, we have talked about temperature, we have talked about the enzymes and substrates being specific. We can talk about the pH. This is also another factor that can be giving us some evidence that it can interfere with the structure and therefore it can enable the molecules to work differently. Now we really know that the enzymes are protein in nature, and the molecules that they are working on will be having specific dimension. So the molecules that we have is a long chain joined of amino acids and the peptide bonds. So these particular peptide bonds, when a structure is going to be formed, we are going to have different shapes, which is the three-dimensional shapes, which can start from the secondary tertiary, and quaternary. Now these shapes, we had agreed that they are always formed due to joining of the bonds, like we can have the hydrogen bonds that we have talked about, ionic bonds, and disulfide bonds. These are some of the bonds that help us in the formation of those particular shapes that we are talking about. And when the enzyme is working on a substance, we have what ranges of the pH that we have. Now each pH will be affecting the type of the bonds that we have based on the hydrogen ions that we have in that particular pH. It will be changing the shapes that we have, and therefore it can interfere with the working of the enzyme. Like for example, we have got some enzymes that only work well when the pH is acidic. Like we have got trypsin, for example, is working well in the basic condition. So we can have a graph showing us this, and then you can see stopping there. So if this is the pH of 8, this is slightly alkaline, so this is trypsin. So this enzyme, it means that it works well in the pH of this, and this one is due to the shapes that are going to be formed. The hydrogen ion that is in that solution is really affecting the shape that is coming in. So those shapes are formed well when the pH is 8, and therefore these enzymes out of this particular pH, it is not going to work very well because of the bond formation of the new structures that are formed when the molecules are reacting with one another. That is one thing that is worth noting. Equally, we can have some enzymes like the pepsin, which work well when the pH is more of acidic. So when it is beyond acid, you will realize that the reaction will be going down. So this is the optimum pH, which in this case is 4. So this is pepsin. So you can see we can have ranges of enzyme based on which pH of the substance that we are having that it is working with. So that is one thing that is very clear that we should be noting in this particular case. So that is what I really want us to understand in this particular thing. So it is clear for us that the pH, the temperature, will normally try to affect the rate at which the enzyme is working, and also the quantity of the substrate. If the enzymes are constant, they'll be telling us which are the molecules that are going to come out at the end of the day. So it is really clear, and it is worth noting that we should be looking at what are the terms that we use when we are referring to the exam. There are those particular exam words that we should be keen with, so that we'll be bringing out our point clearly in every explanation that we are making. So that is the end of this particular lesson of the protein, and the example of the protein that we have looked at is the enzyme. Next time we meet, we'll be talking about the DNA structure, and we'll be seeing how it works with us. So that is the thing that I want you to do. If any case there is a point that you need to clarify for clarification, you can contact, and then we'll see how to help you further. Thank you very much.